Thank you for the invitation. I'm glad to meet Richard, Chris, and Rob again. And let's begin with this talk on plant science data integration, and more precisely on the history of the past 10 years and how we went from building dispersed community standards to defining a consistent data life cycle. So the objective of the plant research communities that has gathered around this plant standards building was motivated by the need of increasing the sustainability of plant production because of the climate change that triggers extreme weather conditions, pests, and diseases. And as a consequence, the fact that the United Nations has designed several sustainable development goals towards agriculture. So to address this need, it is necessary to gather and to strengthen plant production by crossing genome, environment, and phenomic data and information following the FAIR principles, and therefore being able to do plant breedings, in other words, studying genetic variation by traits, or climate change studies, in other words, studying genotype by environment. And the question when we want to do this FAIR integration is first, are we dealing with big data? It's a buzzword that has arrived 5 years ago, but it's still present. And I'd like to make a quick parallel with astronomy. First, big data is defined by 4 V's: volume, velocity, veracity, and variety. For the volume aspect, with astronomy, it's totally obvious that they are dealing with big data because they have hundreds of petabytes of data. But for plant phenotyping, for instance, for the biggest high throughput studies, you can have 10 terabytes of data per study. It gives 2 to 5 petabytes of data per country. It's not that big. Big data for plant phenotyping, for research data, for plant volume is not a problem. About veracity, with plant phenotyping, we've got a question of reproducibility, of provenance. We've got solutions. DFW just before mentioned some of those challenges, and there is an adoption in progress of good practices, including the use of the Pro ontology, but it's still in progress, so it's still a challenge. And finally, the real challenge, the real big data challenge for plant science is variety. We've got multiple scale. We have seen that we've got physical scale with single experiment to phenotyping networks, to even experiment over decades, such as LTE. We've got tons of data formats, images, videos, even sounds or spectrum. So variety is definitely a challenge for plant research data, and especially phenotyping. For the next step of my presentation, I just would like to get back to the history of those past 10 years. 10 years ago, the crop ontology has formalized one aspect of the semantic description of plant phenotyping data. 2 years later, MyAP went one step further, and I will describe later what is the complementarity between those two data standards. In 2015, several communities formalized and gathered to advance both on the standards and on the development of tools to ease data management for wheat and other species. So wheat.is from the wheat initiatives of the G20, Elixir, Emphasis, both European infrastructures. 1 year later, the breeding API set up standards dedicated to computer scientists. 2 years later, there has been at least a good partnership on one official partnership between Elixir and Emphasis. And finally, we have formalized all of this through a roadmap a few months ago. And I'd like to explain how things happened, to begin by the end and by describing to you the Elixir roadmap. So this roadmap has been published in F1000, so feel free to read it in detail after my presentation. And the objective is to build and strengthen a sustainable tool federation. This idea of a federation rather than a centralization is absolutely instrumental in how our communities have worked. It addresses the needs of crop and forest trees, and it aims at coordinate and standardize data management, access, and analysis by facilitating data integration for phenotype, genotype, environment, genetic resources, literature, because there is a goldmine of data in literature, omics, and finally system biology. We are providing means to enhance visibility, findability in the fair sense, and sustainable access to plant research data. We do a lot of training, outreach activities to ensure adoption, development, improvement of standards or community recommendations. And finally, we need to implement this roadmap through projects, both to get coordination for some tasks and second, to get some funding. So this roadmap has been formalized through a series of tasks and use cases, and that forms a classical data lifecycle with a first step on finding relevant data sets, both by creating new experimentation and by gathering existing data, finding them, then processing, integrating them, those data. And by integration, we mean both linking the data, doing a semantic integration, as well as normalizing the data, therefore doing a statistical integration. Then comes the analysis, visualization part, data storage, and finally, data publication and sharing that enables the findability of the result of the knowledge and the data, and therefore enables the beginning of a new data lifecycle. Several tools enable already this life cycle, such as data standards, analysis tools, visualization tools such as Canet Miner, data repositories, and standards again. So those data standards are in fact of three types. There are first semantic data standards, which are about describing the data. It's controlled vocabularies, which mean term names and definitions. And those terms can be linked through semantic links between them. And the important thing is that for them to be useful, for those semantic standards to work, to be adopted, they need to be biologist-driven. They need to be very close to the language of the researcher. And therefore, they are very complementary of structure standards that enable the formatting and organization of the data. They provide data models with fields that take advantage of the semantic standards. And among those data standards, you've got VCF, GFF, MyAPI, etc. And for structures standard to work, they must be both biologist and computer scientist driven. Finally, you've got technical standards, which are purely the responsibility of computer scientists and address purely their need, and that provides data integration and sharing and interoperability between tools and systems, such as GA for GH for genotyping data, or the breeding API for phenotyping. So, some example of phenotype or in general plant semantic standards around ontologies. So there are general ontologies addressing the needs of omics and genomics, such as the gene ontology and the sequence ontology. But there are plant-specific needs And especially what I'm showing in this schema, courtesy of Lawrence Cabrera, is a model that has been formalized by the crop ontology 10 years ago and adopted and announced by Emphasis and Elixir. It stipulates that any variable must be composed of a trait, such as the plant height, a method, total height, first tassel branch, or the highest pixel when you are dealing with images analysis, and finally a unit, therefore forming a triplet. For the phenotype structure standard, the structure standard for plant research data, I'd like to introduce MyAPI. Full information are available on myapi.org. MyAPI standing for minimum information about plant phenotyping experiments because it addresses either single experiment or multi-local network. It is a standard that has been developed by many stakeholders, Biodiversity, Emphasis, Elixir. It's an open community, and I'll come into details of how this community works a bit later. And it addresses the needs of crops and woody plants. It is computer scientist friendly with an explicit data model and semantic representation through an ontology here, and biologist friendly with a lot of work on clear definitions, examples, trainings, outreach, etc. Finally, it's a specification with multiple implementations through a file archive, is a tab, or a semantic representation, as well as web services and a data capture tool. There is also a publication here that gives a full overview of this. Finally, the last member of the triptych is the phenotype technical standard, the breeding API. All information available on brapi.org. It's another international collaboration which has been highly community intensive. It connects data repositories and tools to provide genotype visualization through something, for instance, Flapjack. Tools integrable in any BRAPI compliance system, like the studies graph preview and filtering that is showcasing here, our analysis environment, field data capture, or fair data discovery. The next step is how this MyAPI standard has been extended to go beyond plant phenotyping. Indeed, we want in the research communities to go from plant phenotyping to genotyping to do genetic analysis and breeding. This means that we have data integration constraint by being able to link data from genotype to phenotype, as I already said, and also to link several experiment fields within a phenotyping network. To do this, we need to identify key resources, pivot objects that allow us to go from one dataset to the other. The obvious one being the biological material identification, as well as the phenotyping variables, and to a lesser extent, the location. And the good thing is that MyAPI already encompasses all of those three fields. As a consequence, what we have done within Elixir is injecting MyAPI in the API used to submit data in the EBI databases, especially those used for genotyping data, ENA, biosamples, and EVA. And thanks to this submission, which is now MyAPI compliant, we have the possibility to get breeding API compliant datasets and therefore index them in data portals such as FEDER and integrate them with phenotypic information coming from other data repositories.
I'm sorry, if you can start to conclude, because otherwise we're not going to have any time left for discussion. Thank you.
Yeah, okay, very sorry about that. Yeah, maybe just the one thing I'd like to say about the communities that has built MyAPI and BRAPI, there have been huge success And this is thanks to technical and scientific expertise and the existence of pragmatic use case. It is thanks to fundings coming from European project to Gates Foundation for event fundings, travel, in-kind, hosting institute, etc. We needed coordination, community management, governance equity through neutral leading instance, through agreed boards, democratically elected. We needed a lot of listening, of discussing. All the decisions has been made through technical consensus with a leader, either a single coordinator or a review board or a steering committee that has the legitimacy to make the final decision after the discussion. And finally, by closely coordinating this work with other standards. Just one thing, for dissemination, beyond training, there is a need to build standard registries such as fair sharing or the WIT-IS data standards that has been built over a few years. You've got here the reference of the papers that describe it. We have also made a lot of effort of integrating the data standards in tools to ease data capture, including FairdAMSIC or COPO, which are all MyAPI and ESA compatible. And finally, to address the findability challenge, we have developed data portals that capture metadata, that index metadata from any data repository around the world and enable their searchability in a centralized search portal named FEDER. As a final words, the advantages of building those data standards, even if they are highly time-consuming, is that they enable long-term data reusability. It enables the possibility to capitalize on existing standards, to extend them. Then we need a lot of community management, a lot of training. We need to have a few full-time persons in charge, and the more people on board, the better. And with those final words, I'd like to thank you, and sorry for the